Okay, in this series of short videos, this video is on peptide bond formation, the challenges. This is the usual disclaimer. So, peptide bond formation during protein biosynthesis is thermodynamically unfavorable. So, that bond formation cannot follow the laws of thermodynamics during the translation. And as a result of that, this becomes a bottleneck. This unfavorable thermodynamic situation, it is overcome by pre-activating the amino acids. The building blocks are pre-activated so that the reaction becomes thermodynamically favorable. So what does it mean? It means that during protein biosynthesis, the building blocks are not free amino acids. They are not free amino acids. Rather, they are amino acid esters of tRNA, where amino acids are esterified either to 2-hydroxyl or 3-hydroxyl of terminal A of the CCA at the 3' end of the tRNA. So, free amino acids do not participate in the protein biosynthesis, rather they participate as esters of tRNA, where they are esterified to either carbon number 2 or carbon number 3 of the terminal A of the CCA. So this is terminal A of the CCA at the 3' end of the tRNA. So this is the inverted tRNA. You have the A, terminal A, okay, and amino acids are esterified either to carbon number 2 or carbon number 3. This is amino acid. So building blocks are amino acid esters of tRNA and not free amino acids, wherein amino acids are pre-activated. This reaction is catalyzed by specific amino acid tRNA synthetases. These are specific enzymes. They are called as amino acid tRNA synthetases. So these amino acid tRNA synthetases, they are discovered by P. Zamachnik and M. Haugland back in 1957. They are also referred to as activating enzymes. And they catalyze the reaction in 2 steps shown here. In the step 1, amino acids react with ATP, so energy input is required. So you get amino acid AMP with the release of PPI. And in the subsequent step, the amino acids, the activated amino acids are transferred to tRNA. They are esterified to carbon number 2 or carbon number 3 of the tRNA. So what you get an amino acid ester of tRNA with the release of AMP. So there is at least one enzyme for each amino acid which participate in protein biosynthesis. Thus, accordingly, there will be at least 20 enzymes, and these 20 enzymes, they are classified into 2 subclasses. In class 1, the enzymes, they catalyze activation of amino acids with larger or hydrophobic R groups. Another important thing is that amino acid is esterified to carbon number 2. Class 1 enzymes will esterify the amino acid at carbon number 2 of the terminal A of tRNA. The activation domain of the enzyme has a parallel beta domain. This is a structural difference between the 2 classes of enzymes. So one is, in case of class 2 enzymes, let us see this, they catalyze activation of amino acids with smaller R groups. So class 1 enzymes catalyze activation of amino acids with larger groups, R groups. Here they will catalyze activation of amino acids with smaller R groups. And the amino acids are esterified to carbon number 3, 3OH of the terminal A of the tRNA. This is the second difference. And the active site has an anti-parallel beta domain. Here it has got a parallel beta domain. So these are the 3 major structural and functional differences between class 1 and class 2 enzymes. The amino acids which are activated by class 1 enzymes are given over here: arginine, cysteine, glutamine, glutamic acid, isoleucine, leucine, methionine, tryptophan, tyrosine, and valine. And the amino acids which are activated by class 2 enzyme include alanine, asparagine, aspartic acid, glycine, histidine, lysine, phenylalanine, serine, proline, and threonine. So you can visualize that these are the amino acids which have relatively smaller R groups, and these are the amino acids which have relatively bigger R groups. Now, these amino acid tRNA synthetases, or what we call them as activation enzymes, they also perform another important function. They have, like DNA polymerases, inbuilt proofreading activity, and as a result of that, they enhance the fidelity of protein biosynthesis. Because addition of amino acid to a growing polypeptide chain and formation of a peptide bond is a point of no return. If the incorporated amino acid is a wrong amino acid and a peptide bond has been formed, then it is a point of no return. Then that wrong amino acid will go into the polypeptide chain. So what nature has done, nature has conferred these enzymes like DNA polymerases proofreading activity. So what do? They, in fact, they are specific for both amino acids and its corresponding tRNA. Both the things are important, not only the amino acid, but it has to be a cognate tRNA as well. Now, discriminating between different tRNAs is relatively easy. So because tRNAs can be differentiated based on the anticodon that they have. Okay, they have different nucleotide sequences. But discriminating between two similar amino acids can be challenging. So we have amino acid, for example, valine and isoleucine, where there is a difference of only a methylene group, CH2. So discriminating between these two structurally very close, very similar amino acid can be challenging for the enzyme. But it does it, and what has been found out is that these enzymes, they have a pair of sites. Besides the activation site, the reaction that we have seen in the previous slides, they have an editing site. So with the help of this complementary site, the enzymes are capable of discriminating between even very structurally very closely resembling amino acids. So for example, this is the activation site, okay, where activation of the amino acid takes place in two steps, and they are esterified to tRNA, the cognate tRNA, either to carbon number 2 or carbon number 3. So this activation site has a characteristic that it rejects amino acids which are larger than the correct one. Okay, amino acids which are larger than the correct amino acid, they will simply not be able to enter this activation site. So they are rejected in step 1. The smaller one, smaller than the correct one, they can enter, they will get activated also, but then you have a complementary site called this editing site. What does it do? This editing site hydrolyzes amino acid which are smaller than the correct one. So larger cannot enter over here, and smaller, those who can enter here and gets activated, will be hydrolyzed over here. They will not be allowed to enter into the process of translation. So these complementary pairs of sites function as a double SU, ensuring very high fidelity during protein biosynthesis. Thank you.